Good afternoon all. Today the Samvaad Talk series themes continues on model of computational thinking. Today we have our guest speaker Professor Janaki Balakrishnan to give a talk on dynamics of complex behavior in natural systems. Professor Janaki Balakrishnan heads the complex system program at NIAS, Bangalore. She has obtained a PhD from theoretical high energy physics from University of Delhi. She has subsequently worked and published in a wide range of areas covering many diverse areas of physics, ranging from quantum field theory in curved spaces, theoretical high energy physics at dynamical systems, and biological physics. She was earlier a faculty member of physics, School of Physics at Central University of Hyderabad, and has held visiting and other positions at various places including Institute of Mathematical Sciences Chennai, CARE Bangalore, CMMA-CS Bangalore, JNCASR IISc, Raman in the Research Institute, etc. She is also a guest faculty at Indian Institute of Science, Bangalore. We welcome you, ma'am, for the talk.
Thanks for the introduction. And first of all, I'd like to thank you for inviting me here, and especially Professor Jaya. So I'll use this as a pointer. So today I'll talk about some of the work we have been doing for some years on modeling complex behavior in natural systems. So natural systems ranging from small to the large scales. Okay. So as I mentioned, complex systems arise over all scales. In fact, all the systems we see around us are complex in nature. There is hardly any system you would find which is actually simple in nature. One of the examples I can give of a simple system is that of spring mass system, simple harmonic oscillator. Okay, that is a simple system because the interaction is linear. All other systems we see around us have a nonlinear feature in them, and so they all behave in a complex manner. So these are some examples of complex systems in different scales. So you see here complexity arising in cellular at the cellular scale. So we see complexity in hearing. We have neurons firing, and we are all made up. Our nervous system is made up of networks of neurons. All these cells show nonlinear behavior. And then we have pattern formation in nature, pattern formation at different scales. So here you see beautiful patterns and designs on butterfly wings. There are different kinds of leaves which show patterns in them. You have stripes and spots on animals. Seashells show patterns. You have patterns on larger scales such as sand dunes. So all of these arise, all these patterns arise because of different kinds of nonlinearities. Okay, and you have fingerprints. So you have fingerprint. Each person has a different fingerprint structure, and each of it is because of a different kind of a nonlinearity when you write down equations to model them. Also, you have you have all smaller systems interacting with each other, giving us emergent behavior. So we can model ecosystems. We can model convection in the atmosphere, turbulence. All these are nonlinear phenomena. So here we have illustration of an ecological system having different trophic scales, and all of these organisms are interacting with each other and with the environment. Okay. Also, we see fractal patterns in nature, snowflakes. We see fractal patterns on leaves and many other structures. They are all self-similar in nature, and all of these come from nonlinearities. Here are some more examples I mentioned. So as I mentioned, complex systems are all nonlinear. These are some of the other phenomena. You see repeating seasons or circadian rhythms, biological rhythms, sleep cycles, okay, and cardiorespiratory interactions, geophysical motion of the phenomenon of earthquakes, or you have collapse of buildings occurring sometimes when you have strong winds blowing. Okay, and you have sonoluminescence, which is emission of light because of the incidence of ultrasound in fluids. And then you have all your electronic circuits, starting from the simplest diode. All of these are nonlinear in nature. They all show nonlinear behavior. Even the boiling point of water, if you have the PT diagram, it's not a straight line. You have a nonlinear curve. Okay. So essentially what is common in all of these, all nonlinear systems, none of them obey the superposition principle. So I've shown that here. Simplest examples, the quadratic term x1 plus x2 whole square is not equal to x1 square plus x2 square. Okay, so this superposition is not obeyed. Okay, in order to model any of these, so what do we want to model? We want to model the dynamical behavior, how such systems evolve in time, how such emergent phenomena arise. Okay, so this is dynamics, evolution in time. Okay, so dynamical systems are basically of two types: flows and iterated maps. Flows are systems which show continuous time behavior. Okay, systems continuously evolving in time. They are described by differential equations, which can be ordinary differential equations or partial differential equations. Okay, partial differential equations examples: wave equation, heat equation. Okay, you have evolution in time and another variables such as space. Okay, whereas ordinary differential equations are those which show where you study time evolution of an object, just the time evolution of an object, or evolution in some other variable, just the dependent variable is just one. Sorry, independent variable is just one. Okay, and then you have iterated maps. What are iterated maps? They are systems which which have time evolution in discrete time. Okay, suppose you measure temperature or precipitation today, and then you have the same make the same measurement tomorrow or after some days, but it is not continuously made, it is after an interval, but then you have a measurement, repeated measurement. Okay, so this is a discrete time evolution you are describing. Okay? That is an example of an iterated map. These are also called difference equations or recurrence relations. Okay. So in order to so what do you see in all of these? You want to study its evolution or flow in time, something changing in time continuously. Okay, so as a system evolves in time and you write the differential equations or whatever difference equations, you will see that at certain points in the time evolution, the system does not show any movement. That is, there are stagnation points or equilibrium points. The system stays still. There is no flow at that point. These are the equilibrium points, or they are also called the fixed points. So when you study a system, the dynamics, it is important to study the behavior of the system around the fixed points. You perturb the system close to the equilibrium points. What happens is the nature of the equilibrium stable or unstable, or what kind of behavior does it show. Okay, that gives you a lot of information about how the system is going to evolve in future. Okay, so that kind of analysis is made very locally around the fixed point. This is called a linear stability analysis, and that is a basic tool we use in dynamical systems theory. I'm simply mentioning some of the the ideas used in the subject. Okay, it's a very vast subject, but just the basic ideas so that you have a feeling of it. So equilibrium points are essentially of these types. You have attractors or nodes. You have repellers. These are also nodes, repelling and attracting nodes. Then you have saddle points, which attract in some directions, repel in the other. Then you have center, which shows neutral stability, such as spring mass system, or the pendulum, which is oscillating for small angle oscillations about the pendulum, undamped pendulum. That gives you a fixed point as a center. You have focuses, where if the system is dissipative, it will have a focus, or if energy is pumped into the system, okay, even in that case you will have a focus. So these are different kinds of fixed points, and when you look at the system, you look at an abstract vector space called the phase space of the system, where you can see the evolution of different variables together at the same time. So the system it's parameterized by time, and this is an example of a phase space, and this kind of figure where you are having an illustration of different kinds of fixed points, you can see at the same time that the system is having an attractor, it has a saddle point, it has a repeller. You get an idea of how the system is going to behave at the same time. This is called a phase portrait. Okay, so as for any system, as the system evolves, you will have parameters in the system, and the parameters can take different values. So as the parameters are varied, the system will show a change in the stability. Okay, so at certain points, certain values of the parameters, a nonlinear system can show sudden change in its behavior. So if it was not doing anything, it was quiescent, then at the bifurcation point, suddenly it can start to oscillate, or it can show some other different behavior, completely different, a drastic change. So such a change is an instability in the system, and it is called bifurcation. So a bifurcation actually occurs at critical values of parameters, and it involves a change in the equilibrium of the system and in the topology of the phase space, the dynamics changes. So there are different kinds of bifurcations. These have been classified. Here I mentioned just a few local bifurcations. There are also global bifurcations. There are several other local bifurcations also. Okay, so these are some of the concepts which are used in the study of dynamical systems and whatever I have done as well. So now, why am I mentioning instabilities, bifurcations? Because when you describe any phenomenon, any interesting phenomenon arises because of the presence of at least one bifurcation. There are several bifurcations involved. Okay, and study of bifurcations or instabilities can help in different respects. Okay, for example, when you look at stability of structures such as bridges, then if you knew about the stability of, if you knew about the dynamics of the system and what instabilities are going to occur, then it can help to avoid bridge disasters. So here is an example of bridge collapse which occurred in 1940 in US. This is a very famous example called Tacoma Narrows Bridge, and it collapsed because the structure was not, the bridge was not structured properly enough to withstand the, there were strong winds blowing, and because of the strong winds, the bridge started to oscillate and then it eventually collapsed. So let me show you the video of this. So you can see here, so you can see here, this is a particular kind of instability, particular bifurcation, a flutter instability, a Hopf bifurcation it is. So you can see that the bridge is oscillating very rapidly, and then as the amplitude of these torsional vibrations, they keep increasing, and then the structure of this bridge cannot withstand anymore, the thing collapses. Okay, so a similar thing happened to the Millennium Bridge in 2000, the year 2000, over the Thames River, and that happened because of a slightly different phenomenon, but there also the similar kind of instability occurred. So the study of this subject is useful and very important. Okay, so the same kind of bifurcations can also govern behavior of different other kind of systems at different scales. So for example, here I have mentioned the neuron. Neuronal behavior, neurons fire action potentials, okay, they spike, and then that is how the electrical signals, impulses are transmitted from one part of the body to the other, and we are able to have all these, the nervous system functions because of that. And what causes this firing is because of bifurcations, different kinds of bifurcations, okay. It's not necessary it has one kind of bifurcation for all neurons, different kinds can be there for different neurons, okay. Okay, so essentially study of these, of dynamical systems is useful in biomimetics, where you study the structure and function of biological systems, and because you use them as models in order to design and engineer different, you can design materials and machines, so you can copy, try to mimic how a real biological system is behaving and try to adapt it to a system you are designing, okay. So a simplest example of this is that of Velcro. So Velcro, actually the idea for that came from a biological system, because the inventor actually looked at burrs, those are on plants, some spiky kind of structures on seeds of certain plants, which stuck to the inventor's dog, and he was trying to remove it, and he got the idea of inventing Velcro because of that. So that was a simple example of mimicking biological systems. Similarly, you can make sensors. So if you want to detect sound, or you can have sensors for olfaction or taste, smell or taste, or various other things. So the best sensors are the natural ones, and I mean, even if you try to mimic it, you cannot equal it, right? And so you need to study the actual biological system and model it, and then try to mimic it. Okay. So essentially, in my talk, I will talk about natural systems. I'll give a very brief kind of outline of some of the work I have done. Natural systems at small scales, such as the hearing system, how the ear detects sound, modeling neurons, and then I'll come to natural systems at large scales, where I will talk about modeling ecological systems, and then I'll briefly outline other kind of work being pursued in our group in modeling natural systems. So let me come to the system of hearing. So the ear is the most wonderful sound detector, and it has several features, and all of them arise because it's a nonlinear device. So it shows selective response to frequencies. We can hear different frequencies separately. Okay, we can amplify, the ear can amplify whispers. We can hear, it has the ability to amplify whispers, and we can hear it louder. And then we can hear sounds which order by several orders of magnitude. They vary by several orders of magnitude. 0 to 140 decibels, we can hear the loudness. And then the ear shows another feature. The ear is actually active, and there are sounds being emitted from a healthy ear. Every healthy ear emits sounds of its own. These are called autoacoustic, spontaneous autoacoustic emissions. And actually, spontaneous autoacoustic emissions arise from the ear of any organism having an ear. Even the mosquito, it emits sounds. And each of these sounds are, it is something like a signature. Just as your fingerprint is a signature and your iris is one, the sound which is emitted from the ear is also a signature of an individual. For humans, this occurs in the range 1 to 2 kilohertz. For lizards, it occurs in a different range. For frogs, it's in a different range, and so on. Okay. So the ears actually contain active force generating elements, and this was first suggested by Thomas Gold in 1948. So in all of these, this is a spontaneous autoacoustic emission spectrum of different organisms, and in all of these you see there are peaks occurring in the spectrum. Okay. So the origin of these peaks was not known. So I did some work previously, it's an old work, where I discussed the dynamics of, to understand these occurrence of these peaks. So essentially, how do we hear? So sound enters the ear, and it is transmitted to the inner ear through the eardrum, and it is detected in a spiral shaped structure called the cochlea. And okay, these are the details of the structure of the cochlea. Inside that, and if you open out this cochlear structure, actually when you straighten it out, it is actually, you see there is a basilar membrane. That thing is called a basilar membrane, and on it there are several specialized cells called the hair cells. So these are the hair cells, and these are the ones which detect sound. Okay, so these are aligned all along the basilar membrane, and each point detects a different frequency. So it is like arrangement of keys on a piano. Okay, each is playing a different frequency. Similarly, each in the ear is detecting a particular different frequency. This is an image of this specialized cell, which is called the hair cell. It's called the hair cell because it has some fibrous structures on top which give it an appearance of hair. It's not really hair, but this is called the hair cell. This is called the hair bundle. Okay. So when the sound enters the eardrum, this basilar membrane vibrates. There is a fluid inside it and so on. A lot of details are there. I'm not mentioning that, but it vibrates up and down. So this is the arrangement I spoke about in the cochlea, and the hair cells towards this opening of this ear. These detect high frequencies, and those at the other end inside, they detect low frequencies. Okay, so this is called a tonotopic arrangement in the cochlea. So each sound which enters the ear initiates a traveling wave along the length of the cochlea. So let me just show you this animation. Okay, so I want to unmute this. So this is the cochlea, which if you try to open it out, and this is the basilar membrane, and actually there are hair cells all lined up all along it, and every frequency is heard at different points on this. Okay. Yeah. Yeah, so if you look at every hair cell, actually, you see that in the absence of sound also, absence of stimulus also, these hair bundles on top, they move on their own. They show spontaneous activity. This means that there are some force generating elements which make this movement possible inside the ear. Okay. And these actually are responsible for this spontaneous autoacoustic emission, the sound which we are emitting from our ear. The loudness level is so low, that's why we are not able to hear it. Otherwise, we'll be hearing everybody else's sounds coming from every ear. You can't hear it because you can only hear it if you put a very sensitive microphone inside the ear. Okay. So it is possible to model the movement of the hair bundle and to take all these different components, I mean all the different interactions into account, and then you can write equations for the displacement of the hair bundle and other things inside in the system. And equation is quite complicated. Here I have denoted shown you just some of the linear elements in that, and it is possible to do a stability analysis and to see if we can get any information out of this. Okay. So there are several parameters in the system, such as the length of the hair bundle, its stiffness, so it's something like a spring, a stiff spring moving. Okay, so various things. It's also immersed in a fluid, so there is a drag coefficient coming in the picture, which it's not shown here. So as the parameters change, the stability of the system changes, and it turns out that the system actually operates close to an oscillatory instability, which is a Hopf instability. If you tune the system to work close to that instability alone, the ear shows such properties. It is able to amplify whispers. It is able to show spontaneous movement of the hair bundle. It is able to respond selectively to frequencies. It is able to detect sounds of all these different loudness levels. It's able to do that only because it operates close to this kind of Hopf instability. It's an oscillatory instability. Close to below the critical point, the ear shows quiescent behavior, and just above that, it is oscillatory. So the system is actually poised to the edge of this Hopf bifurcation. This is not resonant. It has a resonant. Each of the hair bundles, it's like a mechanical system. Each of this is detecting a particular frequency. Okay, so so it's necessary to tune. So sound is coming in, right? Sound is the external frequency. So it is possible to tune. So hair bundle, there are several 16,000 of them. So suppose one of this is in 1:1 resonance with that. Okay, then it is the same frequency as the incoming sound. So that is a resonant system, 1:1 resonant. Of course, you can have other ratios as well. Okay, so that is a resonant system. Okay, but that is a resonant system. That is the frequencies. Yes, amplification is there. Amplification, why is it occurring? It is only because the amplification here is nonlinear in nature. It is occurring because, see, you have this is actually the amplification. You can show that the gain of the system goes as See, this is minus 2/3 power of the input. Okay, so the amplification is, what shall I say, it's a nonlinear device, and this power law comes only because it is close to a Hopf instability, otherwise it is not. The instability is a dynamical one. Resonance is a different feature, it is just a matching of frequencies. Yeah, that's right, that is the thing. So how does it remain? That is because of several things, because of the fluid basically in it, in the ear. This is called the endolymph in the cochlea, and so you have a particular concentration of different ions in it, okay, and that also plays a role. Actually, I have not shown the equation, so there are various factors there. It is exactly at that point that only then we are able to hear normally. Okay. No, sound wave is the external stimulus. Yeah, actually the actual thing I'm talking about is concentration of calcium ions actually. There are further details which I have not shown in the hair bundle. When the hair bundle deflection takes place, there are several of these hair, they are called stereocilia. They are connected to each other by thin filaments called tip links. So when the hair bundle deflects, it opens it's a mechanically gated channel, ion channels that are there, and they open out and let in calcium ions. So exactly the amount of calcium ions should come in. And calcium is one of the parameters, important parameters.
That is in responding to the signal, so that you have an electrical signal going to the brain.
That is a different process. This is simply sound detection. It is not detection. It is only the sound coming in. Okay, so once the sound comes in, this has to be interpreted by the brain, right? So when the hair bundle deflects, some amount of calcium ions are come into the hair cells, and because of the influx, that triggers the release of neurotransmitters. There are efferent and afferent neurons connected to the hair cells, and the neurotransmitters are triggered from there, and nerve endings are there, and that is connected to the auditory nerve. Right.
I was wondering what the possible
Right.
before the, let's say, infestation happens, the system on the health cells are in a stable situation. And when the sound cells stay, it hits the brain, it went into an oscillator.
No, actually,
Bifurcation
no. Actually, what happens is, see, I told you that the system is, even in the absence of sound, it is active. Okay, so actually in the absence of sound, there is spontaneous activity. So it is these ions, calcium ions, these are the parameters which poise it to the bifurcation point, and that's why it is active. Now, it is already near the bifurcation point, and now the sound wave comes. Yeah, so now the sound wave comes, and then the bifurcation is disturbed.
the bifurcation happens then.
Bifurcation,
The sound comes.
it might go even beyond the bifurcation point, but it is exactly at that point already. Yeah. So when the sound wave comes, you have an external forcing term, and therefore the bifurcation is, what shall I say, it's no more there, because it has gone beyond that.
To understand that question, whether it is simple resonance within
It is not resonance alone.
if it is bifurcation implies that this change in parameters, the stability parameters is changing. So in that bifurcation, you have essentially a unit cycle capacity.
Correct.
So I was wondering what is it that is going on, which pushes it. It is very close to bifurcation point, but it is just pushed, and then it responds to this possible.
No, even without being pushed, see, you have spontaneous oscillations. What happens? Why are spontaneous oscillations? Are because the system is on a limit cycle rate. That is what. Otherwise, how will it? Because self-sustained oscillatory behavior is what is limit cycle behavior, right? That is already when the system is showing spontaneous oscillations. And now when you have an external sound, that point is disturbed. Okay, and then you calculate the response of the system. It's possible to do that. That's not simple resonance. Okay, so here, using our methods, we looked at a generic system, generic Hopf oscillator, in the presence of fluctuations, external fluctuations or random noise. And then we found that, so this is what we see from our theory, these spontaneous autoacoustic emission spectrum, we see that is qualitatively similar to what is actually experimentally observed. Okay, so similar to understanding the hearing phenomena, we can also try to model other kind of sensory systems such as taste and smell, but these are different from the ear because here we have a chemosensory system. So this was done with Ashok in 2010. Okay, so the other aspect is on modeling other sensory systems such as neurons. So one of the what shall I say, motivations for doing neuroscience or studying neuron dynamics is when you see people showing supernatural, I mean superhuman mental powers. So these are some examples. You must have seen Discovery Channel, and these people were featured there. And some of these are, the first person is no more. He could remember anything he studied. This Daniel Temmet saw the shapes, saw numbers as shapes in shapes and colors, and he could calculate many things. So different people had different abilities. Stephen Wilshire could draw from memory what he had seen just once. These are pencil sketches, not photographs drawn by him from memory. Okay, so these people have extraordinary mental abilities. They are just some motivations I mentioned, not really connected to what I have done. So why do we want to understand neuron and how they behave? So what is the relationship of different patterns of interconnections to different types of behavior? How are the neurons and the connections modified by experience? What are the mechanisms by which all of them produce similar kind of signals? How are they connected? So these are some of the issues in neurophysiology. And so, in the human brain, it has about 10 to the 11 neurons. There are lots of numerous kinds of neurons we have, and they all connect with each other through different electrical or chemical synapses. And so it's possible to model all of them. Basic thing they do is produce an action potential, which is a spiking behavior. So this is an action potential. In order to model just this single spike, it is, how do you do that? Because a nerve cell is a biological structure. It has ions coming in and so on. So Hodgkin and Huxley were the first to model the neuron. And this work was started in 1948, 1952. There's a series of papers, and they got a Nobel Prize for this work in 1963. So they modeled the neuron with a set of 4 coupled differential equations. And there were a large number of parameters in the system. So it's a system which is, even today, this model is the one which produces best the action potential. It is the best model for that. Okay. But then it is difficult to handle because so many equations are there. So there have been a lot of people who simplified the models which worked in different regimes. Some of these are listed here. And beyond that also a lot of people have, you can make your own model. Okay, provided, you have to have some, satisfy some conditions. So now when you work on spiking of neurons or the patterns they produce, you use these models and couple them together, mathematical differential equations. So now what happens is that if there are 2 neurons in the same region of the nervous system, same physiological region, and you give it one stimulus, it turns out that it's not necessary that both of these neurons will necessarily respond similarly to that. Each may show different behavior. This is an example. This is the stimulus which has been given to 2 different neurons, and each of these, although they have the same electrophysiological features, they have responded differently. That is because they have different dynamical mechanisms in them. That is, they have different excitability mechanisms present in them. Okay, so now essentially neuron firing patterns are of 2 types. They can produce spikes or they can produce bursts. Bursts are these which are a series of spikes which are separated by a resting period. These are bursts. bursts are of different kinds and they've been classified, different shapes and types. Okay, and for all of these, there are different kinds of bifurcations which are responsible for producing them and for ending them. Okay, so this is just some work we did recently. I'm just mentioning the kind of, you can use dynamics for modeling different systems. This is also a natural system. This is an image of a pair of neurons near the brain stem. Another work we have been doing recently is on modeling behavior of, this is behavior produced by a single neuron, which is producing bursts, not couple neurons, but single neurons. In this model, we are able to produce in different parameter regimes a large number of burst types. Normally people cannot do that with one model, so we are able to do that. Okay, so these I was discussing modeling systems at small scales, but now we come to modeling large scale natural systems such as ecological systems at large scales. So here in this I will mention.
One question on this.
Yeah.
Neurons part. I'm sure you are aware of the interest these days in neural networks and how these networks. Can we use these dynamic models to understand why neural networks are versus models?
Yeah, you can, I guess. See, these are models of just 2 or 3 coupled neurons. In a network, you have numerous, right? Yeah. In order to understand their behavior, why they behave in different ways. Actually, if you look at, the actual way to study is using dynamics, and why different kinds of behavior, clustering behavior, synchronization, different kinds of properties, or I mean, why these occur? They occur because of different bifurcation mechanisms. But these have not been studied well enough. Networks. Yes. Yes, it's a very complex system. It's difficult to do. But that should be the right way to do it. But there have not been many studies to do large scale systems, networks using dynamics. There are studies ongoing, but not comprehensive enough. Okay, so here I'll describe a work we did in a series of papers on modeling insect pest outbreak cycles. Okay, so here this is a system actually. You know, there are coniferous trees of different kinds. I'm talking about a particular one called the larch tree. So the leaves are essentially needles, large needles. So now these trees grow in cold conditions at high altitudes, very high altitudes on mountains. Okay, so they grow all over the world. There are many large forests in Europe, especially in Canada, in the US, and so on. Even in the Himalayas. In Himalayas, we unfortunately don't have data. I looked for data for our own country, but there's just no data available. Okay, because of some problems. Now, in Europe, actually, there is data on large forests for several centuries. Okay, so that's why we studied this. So it is observed that for many centuries, once every 9 years, it is observed that all the leaves are gone. They're just finished every 9 years. Okay, then they regenerate again. They grow and regenerate, and then after 9 years, again they are suddenly gone. Okay. So they're defoliated completely once every 9 years. So actually the reason for this is because of infestation by an insect pest, which is called the larch bud moth. Okay, it's a moth. Okay, but it eats all the buds. That is as soon as the foliage starts coming on the leaves on the larch trees, as soon as it comes, it feeds on that. Okay, and then completely destroys it. That's why it's called the bud moth. Okay, so these photographs of the large forests growing on the mountains in Switzerland in a region called the Engadine Valley in the absence of bud moth infestation. So you see here it's completely green. Okay. Now the same region has been photographed at a later time. So this is taken in 1972 and another time here, and you see green, it's a brownish color. Okay. So what happens is these bud moths, they completely infest these and wastefully eat these leaves of the large trees, and giving it a scorched and burnt appearance. Okay, completely all the greenery is gone and it has a brown burnt appearance. So this is a picture here of a large tree where the leaves have completely been eaten up after the infestation. Okay. So how do you know it's exactly 9 years after between every infestation? Because these have been recorded for 1200 years in this region manually, and also from you know, these what shall I say, these tree ring analysis. Okay, so tree ring analysis shows that once in 9 or sometimes 8 years these bud moth infestations occur. So they have a particular signature and it's possible to identify that. Okay, so that time series is available for 1200 years. Okay, so everywhere in the world these occur in cycles after some period of time. In the French Alps also it occurs after periodicity. Okay, but the strange thing is, it was observed that it is known that close to Engadine Valley, this is in Switzerland, Engadine, and 1000 kilometers away from it are the Tatry Mountains in the Carpathian region, Carpathian range. So the separation is 1000 kilometers, but here also there are large forests, but here there are no, the infestation is not cyclic. Okay, whereas in the Swiss Alps it is regular 8 to 9 years cyclic outbreak, and here there is no cyclic outbreak. So why is that? Okay, and then the other interesting thing is, all these 1200 years it has been occurring in Switzerland only, every 9 years. So you know that suppose it occurs in 1981, next one to occur would be in 1990. But strange thing is that in 1990 there was no bud moth infestation, and after that there was nothing. So after 1981 there was absolutely no bud moth infestation. Okay, why? So people were wondering, when is it going to come, and why are they are not that they are looking forward to it, you know, they don't want it. But the thing is that large trees have a property, they are waterproof, and they are used for timber. They have, I mean, it has a certain use, and you know, these are very beautiful regions, and if you have infestation, then it spoils tourism. Okay, so that way you don't want to have these infestation at all. But on the other hand, you want to be careful, is it going to occur now or not? Okay, so they were expecting it in 1990, but it never came. So after 1981, it has never happened, and the question is, why hasn't it occurred? Is it going to occur in some future time? So we looked at this question. So it's a question of collapse of the bud moth population cycle after 1981. So in our work we modeled this system, why does it occur cyclically, and we were also able to capture this collapse of this outbreak. Also we were able to explain why this cyclic occurrence does not happen in the Tatra Mountains. So these are the European larch needles, and this is a bud burst of larch. So what happens is in October, this moth, this is actually the, that is the moth which you see, which is what is infesting these trees. So it lays its eggs in October, and then it remains dormant through the winter, and in the spring, as soon as the snow melts and the leaves start coming out, these eggs hatch and these larvae come out, and they start to feed on these leaves, you know, in a very wasteful manner, and then they very rapidly completely defoliate it, and then the entire forest is destroyed. Okay, now this has been occurring in a cyclic way I mentioned. It turns out that in this system there are 3 kinds, basic living things involved. So one is the bud moth, the female bud moth, which is pictured there, and the second thing is its host. It is living on the larch tree, the larch plant, that is its next living thing. It also turns out that there is a third organism which are parasitoids. Parasites which live on the host and kill it are parasitoids. So it turns out there are parasitoids which live on the larvae, okay, and they kill it subsequently. So it turns out all 3 are necessary for these cycles to occur. Okay, so here I have simply illustrated difference between parasites and parasitoids. Okay, so in order to understand this system, so all 3 levels, three trophic levels are required to model the system. Okay, so essentially each is a predator-prey system. Each one is living off the other. Okay, bud moth is eating the large, the parasitoid is living off the bud moth. Okay, now so these are organisms interacting with each other, killing the other, taking its nourishment from the other and killing the other. These are basically predator-prey systems. So this is just a kind of a digression. Predator-prey systems can be modeled mathematically. So if you denote the prey by X and predator by Y, you can model it by a system of differential equations. So here you have birth rate of a prey, death rate of mortality of the other one, the predator. You can also have other terms here. These are some simplistic equations. These are the interaction terms. So one is growing at the cost of the other. Okay. So as the predator population rises, the prey population decreases. As the predator population dwindles, the prey population rises, and so on. So it's a cyclic process. So these can be modeled. Similarly, how much, if you are eating some food, how much can you eat? After some time your stomach will be full, right? You can't eat anymore. Okay. So it's a saturating process. So you can model this, how much consumption of the food can any organism take. Okay. You can model it by equations, and you can have these kind of saturating equations here. These were first modeled by Holling. Okay. So I'm mentioning these things because these are the different kinds of inputs we considered in order to model our system. Okay. Now host-parasitoid interactions were modeled by Nicholson and Bailey long back in 1930s. And because the parasitoid kills the host finally, host will no longer be existing after a certain point of time. The interaction terms are different, they're exponential. Okay. All right. So we considered all these factors. So what are the factors which affect the growth of large trees? So we considered that. So one is that during the vegetation period they should have a lot of sunlight, they will thrive. And then, but they shouldn't have too much sunlight. They shouldn't be exposed continuously to sunlight throughout their life, because then they'll die. Okay, so increased overall temperatures is bad for the large tree and also for the bud moth. And then if you have sparse distribution of the large trees, then it means when the bud moth is there, it has to compete more for the food. Okay, so we have to take that into account. And then you have good rainfall is beneficial for both large and the pest. Okay, so we considered all these quantities, and then we wrote equations to describe all these, the bud moth here, the bud moth, the parasitoid, and then to model the large. What is the thing which we can model for the large? We can model its health. Okay, so the health of the large is modeled by the length of the leaves, the needle actually. The longer the needle, the healthier it is. That's how it is modeled. And so you can write equations for that. So previously there have been attempts to model it, and one of the very important people in mathematical ecology, Peter Turchin, has modeled exactly this system by 2 models, but his models are not giving, they're very finely tuned. If you change the parameters by decimal point, you won't get these cycles. Okay, it was like that. And there were many things it was not explaining. It was not explaining the cessation of these cycles after 1981 also. So in our model, we thought that one of the features which could affect these, all these cycles, and it is the stopping of these cycles, could also be climate change. So we introduced physical parameters such as, 2 climate change parameters, one standing for precipitation and the other which was modeling inverse temperature in these equations. So we had 2 factors here, H and S additionally, and we had changed the terms, different terms we had. Okay, by doing that, we were able to obtain these 9-year cycles. And the model is robust because if you change the parameters a little bit, the thing doesn't collapse. You still have 8 to 9 year cycles. Okay. And so we did the stability analysis and so on. And then we were able to find certain regimes in which we found that some of these regimes could represent the Swiss alpine regions where 8 to 9 year cycles were occurring regularly. And in some of these places, the Tatra mountains, there are no cyclic outbreaks. So there were certain regions such as one on the top which could represent the Tatra region, because there the growth, there is more cloud cover there over the Tatras, and the trees are spaced more sparsely. They are not growing very densely. Okay, so we took that into account in our modeling, and we find that different regimes can represent the different observations. There are no cycles we find for the Tatra region. Yeah.
So the climate change.
Yeah.
So that must have had some relationship to the mortality rate of larvae, right?
Mortality rate of the, no. I mean, we have just kept the climate parameters separately. Just two terms we have placed.
So how does that affect whether
the mortality, we have not modeled the mortality as depending upon the climatic conditions. We haven't done that.
So how did it actually, how was the climate change incorporated?
Yeah, so we had two terms here, physical parameters H and S, one for the precipitation and S for the temperature change. Okay, so if that changes, change of that, it brings the system to a different regime where the cycles don't occur. In the previous models, these terms were not present. Okay, so let me just show you one second. So here we have looked at the stability analysis. We have done that for changing H and S. Okay, and then we see that as they change, the system stability changes. And let me just tell you, and let me finish this, and then because the time is limited. Okay, so now here I have compared what we have done. Our model is shown in blue, and the recorded data in red, and the model by Peter Turchin in green. Okay, so he had two models. This is one of them. So our model compares better with the data than Turchin's model. Okay, so our model shows 9-year cycle as the predominant one, followed by 8-year cycle, and then a 10-year cycle. Then this is the second model of Turchin we have compared with, which his model is in green again, ours is in blue, and the red is the data. Okay, so here again our model compares very well with the data. So our model also predicts the 40-year and 100-year cycles. So we are actually predicted that the next bud moth outbreak would occur in 2021 or 2081. Okay, so why we thought of temperature change, climate change, was because we looked at the climatic conditions over Switzerland. There is a paper in Journal of Geophysical Research. So upper air temperature trends about Switzerland 1959 to 2011. So we can see here, so they published this temperature measurements on the the troposphere and stratosphere, different levels. So you see here over a period of time. So you see at 1980-81, you see a sudden change. Okay, so temperatures are the warming in the troposphere is found to be larger during summer months. So you can see there is whereas stratosphere there is a cooling. Okay, so this indicates climatic change. So climatic tipping point is indicated in the 1980s. Okay, so there had got to be a physical what shall I say, change which occurred the system was affected because of that, and how to incorporate that. So we had put these two terms. Okay, and we have some logical reasons for that, but there isn't so much time. So to explain all that.
So here there are only two entities which are interacting.
Three entities: the parasitoid, the bud moth, and the tree, the plant. Tritrophic system, 3 trophic levels.
No, I was wondering if this ecology is that there's anything else which is entering the picture.
It's just these three things. Of course, when there are three things are interacting, apart from their mortality interactions, you will also have competition between the bud moths for food, for resources. Resources are limited, trees are limited. Okay, the bud moth is also limited in number. So for the parasitoids also, there is a competition. So all these things, and then.
Why I asked is usually in the predator prey.
Yeah.
Theoretic.
Correct.
You only talk about two entities. or at least I suppose a very simple kind of thing, two entities. But then if you take an actual system, there are so many entities competing for resources and trying to escape, getting eaten or whatever it is. So the situation dynamically is extremely complex.
Correct. So actually what you're talking about, just two of them.
I'm wondering if you are missing something by saying that you have a mouse there.
No, I have several parameters in it. I have not described it here. I have actually written it here, but I haven't. The three organisms. I mean, the living system, there are just three. Okay, which are known. It's possible there are others. We don't know. It's possible, but these are the known ones. And the physical interaction you see is with the temperature and precipitation. Of course, there are interactions between them. Like the parasitoid, to search for its food, it's going to take some time for searching. Parasitoid searching time, attack rate, and all these are there. Okay, so those have been incorporated already.
Which are the major?
The growth rate of the bud moth, growth rate of the larch. These are the major factors. Okay, so here we find that unfavorable climatic conditions at Tatras appear to be responsible for the absence of bud moth cycles there, and increased temperatures due to global warming appear to have led to collapse of these 8 to 9 year cycles. Actually, for this system, we can make the orbit diagrams, the bifurcation movies. We can make movies. Normally, these are static pictures. So we can make 3D movies. Actually, we have done that. There's a link to the paper if you click on the scientific reports. So you can see that. So you can actually locate these tipping points. So So one more thing, interesting thing is, it is known that there are 94 species of parasitoids which can affect the bud moth. It is known that 94 of them exist. At a time, you can even have 5, 6, 12. You can have several of them. Okay. But now we did it, the first paper which I just reported for one species. Okay, you can have more than one species, and we wanted to see what happens. Okay, so we find that the presence of more than one parasitoid also affects the cycles of the system. Okay, so previously this had not been done, and we found that there is an alternative explanation for the complete cessation of cycles and non-recurrence of this after 1981, which is the presence of an external another parasitoid. Okay. One more thing is that in the French Alps, on the other hand, the cycles are completely irregular. 8, 9 are not the only ones. There are also 21-year cycles, different periodicities seen. Okay, that is also that explanation also we are able to give using our work. All right. So this was the work on modeling pest outbreak cycles. And now briefly I will tell you just in 1 or 2 slides other kind of work which is being done by our group, which is also on modeling natural systems. So presently we have a DST project for studying the impact of human and environmental interventions on living systems. So what is the environmental impact of, especially of environmental impact of coal mining based activities? That is the focus of that project. So essentially we are studying that in the Singareni colliery region, Telangana. That is the focus area which they have identified for us. Okay. So we have been looking at data, and also we have other aspects of, different other, apart from the Singareni area, other regions also we are studying impact of interventions on living systems. So here climate change effect, global temperature rise has given rise to regime shifts in the climate around the world, and that has produced changes in ecological niches and habitats of creatures, and perturbations have occurred in the balance of balances between different species, and then this has caused change in the amount of resources, food present for different species, and so that has caused these different animals to move away from their natural locations to different places in search of food. And so they also many times interact with humans. What shall I say, human-animal conflict also occurs because of this. Okay. So these are some of the aspects we were required to study in this project. So this is one problem we are looking at in our in my group. So this is the there has been sudden explosion of jellyfish blooms around the world, all around the world. Sudden increase, too much. And even in our country, this has been observed in Kerala, in the backwaters in Kollam region, and also in Bombay, Goa, that region. Okay, suddenly this is some picture of jellyfish blooms in Kollam, in Kerala. And it has been hypothesized that this is because of global temperature rise. There have been several studies. Okay, but that's not the only thing. There are other factors as well. Okay, so for example, throwing a litter in the beaches, that actually causes jellyfish numbers change because of that, because there is more substrate present for the jellyfish to thrive on. And then there is also competition for the same food between the jellyfish and other forage fish. Okay, there are several other factors. Okay, so we modeled this system, and this is a basic food chain for the jellyfish. So you have jellyfish here, different species, which are eaten by the predators. For these are basically the turtle, leatherback turtle, also the yellowfin tuna, and then the jellyfish they prey on plankton of different kinds, and for this same food, not only the jellyfish, but there are other competitors such as other fish, sardine, mackerel in the Indian waters. Okay, so we modeled the system. There are all these jellyfish, and the baby jellyfish are called polyps. So we have also equations for polyps, and we have equation for the predator turtle, and for the competitor fish. 5 equations are there, coupled equations, and also we put in climate change factors, temperature. Because global temperature increase we put in, and this is, I'm not sure if you can see it very clearly, but you can see here, the dark blue is the jellyfish, denotes the jellyfish bloom. This one is the light green is for the competitor fish. As the competitor fish come down, see, they are all eating the same food. So naturally this will thrive better. When the competition goes down, jellyfish will be more. These purple are for the polyps, the baby jellyfish. This yellow one is for the turtle, the predator of the jellyfish. Okay, so we find these are pictures, plots for different global temperature conditions. Okay, I have not given the details here. So we find that as temperature rises, the effect of the jellyfish bloom is more. There's more population increase of the jellyfish. That is one work we have done. And another work, here also I have not shown the equation, just one slide. Now we are modeling animal movement. So using radio telemetry data. So we have written down stochastic differential equations, and we are able to simulate movement of animals. Here we are looking at hornbills in particular in the Pakke Tiger Reserve region, Arunachal Pradesh. So we have done this work in collaboration with the Nature Conservation Foundation in Mysore, and this is he was my project associate until recently. Now he has gone to Chalmers. So you can write stochastic differential equations and simulate their movement, and model works very well. And if you put, there's a river running here, and there are human settlements here. We model that through, through certain potentials in the equation, and we see that the movement is not there. I mean, the animals get repelled from that region. So human interaction actually affects their movement. Okay, and then finally, there's one more work I'd like to mention with another my JRF. So she's not researcher, she's my JRF now. So here she is modeling, it's been observed that if you give too much enrichment, nutrient enrichment to the plants, they actually, after some time, they initially grow well, after some time they decline, their numbers decline, and the biodiversity, other kinds of plants or species also decline. Okay, so is it possible to understand this? So we've been doing some work on that. Pardon? I will tell you after this over. Okay. So in order to let me summarize the results. So we're able to model natural systems at different scales, and it's possible to model phenomena observed in nature through appropriate dynamical models. So you should know which are the parameters you should tweak, and then in the case of pest outbreak cycles, we are able to reproduce the observations which have been seen. Okay, so thank you very much for your patient listening. So to answer your question, the nutrient enrichment, yeah, actually it happens because especially this was investigating enrichment by nitrogen, nitrogen rich thing. Also there are some other chemicals present, and there is also presence of bacteria. Certain bacteria break these up. So the equations involve equations for the bacterial. Some of the bacteria remain dormant and don't act. Some of them act. Okay, you have to model all that, and then you see that after certain amount of time, the thing goes down. The plant species goes down because of bacterial interactions essentially, which they remain dormant after some time. And it's not published, so I'm not saying more details. You can model it because of actually these physical interactions between. No, I mean you always know that too much of a thing is bad for you, right? It is essentially that. Here it is essentially growing, but after some time, because the bacteria can't break up the nitrogen-rich thing beyond a point, some of them go into dormant state, some of them remain active. Because of that, this goes down, decline happens. Yes, yes.
But I'm wondering where's the nonlinear because it seems to be a linear change.
Oh, no. Oh, I showed you in that because see, this was in the pest thing. That was here. See, here you see.
There's a coupling between the later.
Sorry, in this equation. Here. So here you see, this is an exponential term first of all for the parasitoid, and there also in the first one, exponential term, and this one, z upon 1 plus mu z. No, that itself is nonlinear, and again you have the whole thing multiplied by x. It's entirely nonlinear.
And the second one was you were speaking about neural activation.
Yes.
Is the same true for neural pathways as well? Because new pathways are usually formed by neurons. So can that also be modeled?
I think they already are, but to how much extent I don't know. Already there's a lot of work in the literature which also discusses bifurcation mechanisms in networks, neural networks. Yeah, but they don't use like Hopfield models, not that, but neuron models like such as these to model biology, but networks of them. And there are bifurcation mechanisms found which show different kind of behavior after certain points, after certain bifurcation points. Yes, but it has not so much work has been done yet in this area, although there is literature existing.
So you know that in typical engineering systems that we work with, there's the only concept of Hamiltonian, etc.
Yeah, right.
What sort of philosophies do these belong to?
Lagrangian Hamiltonian.
Like do these models belong to any of those classes?
See, of course. See, you have Hamiltonian systems, right? And you have Lagrangian systems. Out of Lagrangian, what do you write? Use the Euler-Lagrange equations of motion. These are equations of motion. Now, what you are doing with the equations of motion, you are simply looking at the stability and how the time evolution.
These are like equations of motion.
Of course, these are equations of motion. What is x dot dx by dt?
But many of these things would be like dissipative elements.
There are dissipative elements, certainly. There is hardly any, in fact, very few Hamiltonian systems are seen, conservative. Very few are seen. Most of them are dissipative. Yes, correct. This is also, limit cycles are there. Whenever limit cycles are periodic behavior of self-sustained oscillatory behavior, limit cycles, this kind of periodicity is because of dissipation only. Dissipative elements. There is dissipation up to some, then energy is pumped in the system because of its nonlinear nature. That is why it occurs, limit cycles.
Can also some kind of a symbiotic relationship between the, kind of a balancing system.
Correct.
And,
Yes, you are right.
Outbreak as some kind of a bifurcation from that point.
See.
So usually when there is no outbreak, there is some kind of a balance.
Correct. Actually, you can consider this to be symbiotic system, and that is correct. I mean, the model is anyway there. Whether you look at it as a symbiotic system or as a in a different way is up to you. But otherwise, also symbiotic systems in between living organisms. There is a lot of literature on that, and you study bifurcations in that. Yes, correct.
Okay, so we'll take the rest of the discussions outside. Giving the feedback. Would like
Thank you so much.
Thank Dr. Janaki for giving.
Thank you very much.